Okay. Some of you guys asked for assistance for the first and only quiz for Unit 6. So I thought I would go over some of this specifically because today we have a virtual day caused by the snow, which I don't know about you guys if you can see out my window, but I got no snow out there. For you guys, it looks pure white. Maybe it does look like snow, but I'm looking at a clear street. So a clear day. We're looking at how traits are passed on from parents to offspring. Okay. How genes control traits. We'll go a little bit more into that a little later next week. But one big question I've been getting asked is, how does meiosis tie in with traits being passed on? Okay. So meiosis, we studied that last unit. That was Unit 5. Meiosis is when you take a cell from an individual that has 2 copies of every single chromosome, because that individual has a mom and a dad, so they have 2 copies of every single chromosome, one from mom, one from dad, and they replicate all of them. So now they have 2 sets of the 2 sets, and then they're going to produce either 4 sperm if they're a male and 4 eggs if they're a female. Okay. So how does that relate? How does that connect to our alleles, our letters, dominant, recessive traits, what traits a kid gets? Okay, if that person were to have a kid. So I'm going to do an example right here. Okay, I'm going to show a pedigree, and let's say I'm talking about a woman. This woman, let's say simply, she has a pair of pretty brown eyes, let's say. And let's say that brown eyes are, we'll say big B is brown eyes. Let's say little b is blue eyes. So big B is dominant, so that means brown. The phenotype, which is brown, phenotype is how you look. The phenotype of brown is dominant. The phenotype of blue is recessive. Okay, and let's say she has brown eyes, and let's say she is what we call a heterozygous individual. Heterozygous means you have 1 dominant, 1 recessive allele. Okay, so big B, little b. Okay, so she's a big B, little b. She does have brown eyes, but she does carry the allele for blue eyes. So one day she's going to have offspring. So let's look at the cell that she has. So she has big B, little b. But we know before she undergoes meiosis, she needs to replicate her chromosomes and your alleles, your genes that code for your traits. So your alleles, okay, your alleles are essentially your genes, the genes for eye color. These genes for eye color, these alleles, need to be replicated. Okay, so big B, little b, we're going to make a copy of both of them. So let's say I erase this now, and now we're going to have big B, little b, big B, little b. Okay, you always replicate your chromosomes before you do some sort of cell division, whether it's mitosis or meiosis. Okay, meiosis, you're going to end up with a specific number of cells at the very end, a specific number of daughter cells. And the specific number you end up with, hopefully you can guess, but you're going to end up with 4. And if you see here, big B, little b, big B, little b, she has 4 letters, she has 4 daughter cells. What's going to happen is during meiosis, during the 2 steps of cell division, essentially one of these alleles, one of these brown or blue alleles, chromosomes, are going to be passed on to each of these daughter cells. So let's say one of them gets a big B, one of them gets a little b, another one gets a big B, another gets a little b. What I need you to ask yourself is, out of all of these, how many are big B? And how many are little b? Well, you could tell yourself whether it's a fraction or a percent. I would say 2 out of the 4 are big B, which is 50%, or 2 out of the 4 are little b, which is also 50%. And so that means when this woman has a child, she's either going to pass on a big B or a little b. Okay, 50% chance for big B, 50% chance for little b. Okay, and so we have 4 of these, but realistically, since it's a 50-50 shot, we really don't need a big B, little b, little b. Since there's 2 of each anyways, I'm going to just erase 2. Okay, I'm just going to ignore 2 of these for now. So what I'm going to do is erase these, because essentially, if we have a big B, little b, we know there's another big B, little b, but if I have one big B, there's a 50% chance that it will be a big B she passes on to her kid, or a 50% chance she'll pass on a little b. If I put a big B and little b here, the chances are still the same. So I'm just going to ignore these other 2 for right now, these other 2 eggs she produces. So that means this woman will pass on either a big B or a little b to her offspring. And let's say she gets married to a man. And this man, let's say he has blue eyes. Little b, okay? The reason you have to have little b to have blue eyes is blue, the phenotype for blue, okay, the allele little b is recessive. And for a recessive trait to show up as a phenotype, you need 2 of those recessive alleles, 2 lowercase letters, 2 little b's, okay? And then what if they have a child? I don't know if the child's going to be a boy, a girl. In this circumstance, it doesn't matter, okay? I have to ask myself, what is that child going to have? Are they going to have blue eyes, brown eyes? What's the % chance of them having blue versus brown? Okay? So, father's a little different. He's not heterozygous. He doesn't have 1 of each letter. Okay? He has 2 of the same letter. Same comes from the root word homo, which is one of our vocab words. He is what we call homozygous. Homozygous. And specifically, he is homozygous recessive. He has 2 of the same letters, and the specific letters he has is 2 of the recessive ones, okay? And let's say we're going to keep track of the blue eyes in the family, because blue eyes are recessive, not going to show up very often, so let's keep an eye on them. So what I'm going to do is I'm going to shade in around this, and what shaded in means in a pedigree is that that's the trait you're tracking. And if someone has a shaded in part of the pedigree, whether they're a box or circle, if their box or circle is shaded in, that means they have that trait. He has blue eyes. And all he can pass on is a little b, okay? Just like how mother can only pass on a big B or a little b, he can only pass on a little b or little b from his sperm, okay? So, how can I figure out what this kid's going to be? I'm going to put them as a special diamond, because I have no idea if they're a boy or a girl. So, mom can only pass on, ignore that, my marker's leaving me. Mom can only pass on a big B or a little b, dad can only pass on little b's, so how do I figure out what's the chances of the kid being big B, little b, little b, what color eyes are they going to have, okay? If you're trying to calculate probability statistics with a gene, with an allele, for an offspring, what their genotype is, and so what their trait will be as a phenotype, you need to use a Punnett square, okay? Punnett squares, super easy. I love them. I think they're great to use, and we don't even get too much in depth where we track multiple traits at once. We track usually just one at a time, okay? So I'm going to grab my marker. Marker's been grabbed. So, Punnett squares look like this. They're a 2 by 2 square. So I'm going to make a square, and I'm going to make 2 columns, 2 rows. And what you have to do is place the alleles of one parent on one side. So if you look at the very top up here, I'm going to put mom up here. Mom has a big B, little b. So I'm going to put that at the very top of the boxes. Not inside, on top. So big B for mom, and then a little b because she can also pass on a little b. Okay? I'm going to put her 2 possible letters, her 2 possible alleles she can pass on. Okay? And I'm going to put both the dads on one side. It doesn't matter really if you put it on this side or this side, as long as you put it on one of the sides. I like to do the left side just because when it comes to reading left to right is easier for me. So I'm going to do little b, little b. Now, inside the box will tell you the probability of a child having a specific genotype, which relates to an associated phenotype. So, each box represents 25%. And you may be wondering to yourself, why 25%? Well, if you take all 4 boxes, and if you add them together, you're going to get 100%. And so if you split the entire box that equals 100% into 4 separate boxes, well, 100 divided by 4, 25%. So how do I split these letters up? Well, you got to look at the perimeter, essentially, of each small box. You need to look on what's on each side. What's on top, what's to the left, okay? So if I look at this box, up top is a big B, to the left is a little b, so that means this box will have a big B, little b. If I look at this box, up top, little b, to the left, little b, this will be a little b box. If I look at this bottom left one, big B at the top, little b at the side, we got a big B, little b. If I look at this last one, little b at the top, little b on the side, little b, little b. So now I have to ask myself, what are the chances of this kiddo having brown eyes? Well, if they have the genotype of a big B, or a big B, little b, as long as they get one big B, the dominant phenotype, which is brown eyes, is going to show up. So there seems to be a big B here, so 25% plus 25%. There's a 50% chance for brown eyes. And then to have blue eyes, just like the father, you need little b. So to have a recessive phenotype, you need 2 alleles that are both recessive, lowercase, okay? So little b. I have little b right here. So I got 2 boxes, 25 plus 25, 50% chance of blue eyes. So 50% chance for brown, 50% chance for blue. Now what I'm going to do is I'm going to make one change to our pedigree, and I need you to decide which is it. Is it blue or is it brown? Try to remember what we're keeping track of. So let's say they had a daughter. I'm basing this kind of off my family because my sister was the firstborn. I came after, and I'm like the 2.0 better version, but my sister came first, so I'll give that to her. Let's say this girl that was born is shaded. If she is shaded, what does that mean? Does she have brown eyes? Does she have blue eyes? That's what I need you to think about. Whip. Okay, that's a little cringe. You gotta sprinkle that in the video. If she is highlighted, if she is shaded in, that means she has the trait we're tracking. And the trait I said at the very beginning that we're tracking is blue eyes, that phenotype, which must mean the daughter also must have little b. So Punnett squares predict probability. Pedigrees will track a trait. Dominant traits, dominant alleles. If you get one allele that shows up in your genotype, so your genotype is your combo of letters, big B, little b, little b. Whatever combo you have, that's called your genotype. Dad had little b, that's his genotype. Mom had big B, little b, that's her genotype. And daughter had little b, that's her genotype. Your genotype determines your phenotype, aka your genes determine how you look. Your phenotype is how you look, the trait that you show on the outside. So, dad has blue eyes, that's his phenotype. Mom has brown eyes, that's her phenotype. Daughter has blue eyes, that's her phenotype. Okay? Genotype versus phenotype. That's one of the main questions I've been getting on the review so far. I'm going to erase this. Goodbye. Okay. With that being said, I'm going to try to make a little review based on proteins and enzymes, because that's what we're going to be diving into for the lab for the rest of this unit. And then we're going to finish up talking about potentially some lactose intolerance, finishing up enzymes, and maybe talking about how DNA ends up making your proteins at the end of the day. Okay? So with that being said, I'm going to wrap this video up. A little chunk of the first part of Unit 6. I'm going to try to cover a little content from the end of Unit 6. Cool. Thanks. If you have any questions, just send me an email. Let me know if you have any other questions on any of the other assignments, preparing for quiz, or preparing for test. Thanks.